Hi, everybody. Welcome to our genetic course. My name is Dr. Saw Sapanian. We will review some of the basic concepts of genetic in this course. Today, we are focusing on learning some of the essential vocabulary, as well as some of the essential overview concepts that are associated with genetics. You will notice that some of the information will be repeated on future lectures, but again, this lecture is acting more as a familiarizing you with the vocabulary as well as giving you an overview of the course. The first few slides of the PowerPoints are always going to be learning outcomes. I would recommend for you guys to spend some time reviewing them directly from your PowerPoints, and then starting the recording and listening to the presentation. So let's go ahead and get started. So what is genetics? So we are taking a genetic course. So let's see what is the definition of genetics. So genetics is a branch of biology that studies the pattern of inheritance, and how those traits that we have inherited have a variation, and how does it effectively transmit it from the parents to the offsprings. So simply put, genetics is the study of genes and the concept of heredity. Um, Now, it is very important for us to understand genetics in general. I think every person should have some basic understanding of the genetics, as it has find its way deep inside our daily life, in terms of medication, in terms of us creating our offspring, procedures such as IVF. Um, and again, a lot of things that have to do with us creating our next generation. It's also a big part of our forensic. It's being used by the police to figure out if a crime has been committed and who actually committed the crime, if there is any relationships in terms of paternity. There is genetic built into the food that we eat, the animals that we grow, the plants that we grow, etc. So it is a big part of our day-to-day life and the policies that we make. So it's important to understand it, and also, I guess, have a generic understanding of that, the concept, so you can make informed decisions in terms of the policies that may come up, and basically better understand what happens in your real life. Now, here, this is a common thing that has become popular in the past few years. It's called consumer genetics. The most common consumer genetics that you see is these type of genetic testing, where it tells you information about you as a person. It tells you what your background is, what your race is, what disease you might be more susceptible to, what medication may work better for you, and if you have any siblings that you may not be aware of. Basically put, it gives you a lot of information of who you are and what type of traits you have and where those traits actually came from. Now, I do want to take a moment to define the word genetics and compare it to the concept of genealogy. And I think there is a misunderstanding in general public where they use these two words interchangeably. So we want to make sure we understand that when we talk about genetics, we're looking at the study of the science of the study of the genes and how those genes gets passed down to the next generation. It's the fact of us figuring out the information, figuring out what genes are, how does one gene gets passed down and what traits do we create by those genes. Yet it be, again, having blue color eyes or darker skin tone, or being susceptible to specific conditions like breast cancer, etc. Now, when we talk about genealogy, we're looking at how we are related to other people. So we're trying to figure out if we are related to, let's say, somebody famous in the past. We're basically tracing our lineage. We're not really looking at the science behind it, rather than using the science to figure out our relationship with other people. And as I have used the word heredity a few times, again, that concept of heredity, we're focusing on how the genetic information and traits are being transmitted from one generation to the next. And also how those characteristics might appear as they get inherited or passed down to the next generation. Is it going to affect one member or multiple members? And again, we'll talk about that in terms of complex genetics and complex traits versus what we call Mendelian genetics, which is based on dominant and recessive traits. Now, because we're talking about genes, let's introduce some of the generic terminology that you may hear that helps you understand and evaluate a statement related to genetics. So if you look at a human being, the smallest units that is independently surviving and basically can sustain itself is referred to as a cell. So a basic unit of life. So a cell can be alive on its own because it has all of the necessary components to make energy, consume that energy to survive. That's what a cell is. So at the bottom, we have a picture of a cell. If we zoom into the cell, we also have this structure, which typically appears as a round structure, easily stained. This structure is referred to as the nucleus. A nucleus is the space where we basically keep our genetic information. And these are the X's that I'm kind of drawing at the bottom. So our genetic information can be very loose inside the cell. And when it's really kind of unwrapped and unraveled inside the cell, they're identified as basically DNA. So it's a double helix, they're very long strands, and also very fragile. So typically, if a cell is undergoing changes such as cell division, what happens to that genetic information? What happens to that DNA? It starts to condense itself, and it condenses itself at multiple levels. The most condensed version of your genetic information is what we refer to as chromosomes. And again, if you look at this picture, here is a typical depiction of chromosomes. Now, we will discuss chromosome structure in much more detail later on. Now, if I look into the chromosome and kind of break it down, chromosomes are broken down to these little segments, and each of these segments are referred to as a gene. Basically, what a gene is, which is the image you have down here, a gene is a instructional manual for you to synthesize proteins, or more specifically for your cell to synthesize proteins. And again, these genes are what you pass down from or you have received from your parents, and you will pass on to your offspring, to your next generation. So this is where that kind of inherited concept comes into place. So again, it is important to note that DNA is condensed to form chromosomes, and those chromosomes have little segments referred to as genes, and genes are what will be encoded to make your proteins or to synthesize your proteins. Now, if I'm looking at all of the genetic information from a person, that full sequence of genetic information, which basically came from two parents, is referred to as the genome. So if we're looking as humans, okay, a typical human, if you're a healthy person with no genetic mutation, you will have 46 chromosomes inside your body. And these 46 chromosomes, basically 23 has come from your mom, and 23 has come from your dad. So if you put all of that genetic information that's basically forming that 46 chromosomes, we refer to that as your genome. So again, you're inheriting one set from each of your parents. For us, that's the 46 chromosome. Now, again, if we unwind this genetic information, which is right here, this is your genome. Imagine it again, received two sets, one set from your mom, one set from your dad. If I unwind this, it is broken down to genes. So starting from here to here, this is basically a depiction of what a gene would look like. Now, if you notice, this is color-coded. So you have areas that are green, right? Right here, and right here, and areas that are depicted in kind of an orange section. So I'm just gonna highlight them like this. So the way we look at this is the parts that are in yellow they're called introns. Introns, there's no point for us. There is no use for us as far as we care about. But the areas that are depicted at this kind of like darker green color, so we have 4 sections of them, they call exons. Exons are extremely important because I can put all of these exons together and use them as the blueprint of producing and synthesizing my protein, which is depicted down here. So all of these little exons are joined together from the gene, form one long sequence, and that is the sequence that is used as a blueprint to form your proteins. Now, if I take my whole genome and count all of the introns in all of the genes, all of the exons inside my genome that makes every single protein, then that is referred to as the exome. So exome is all of the exons within the genome combined together to form this much larger sequencing that is called exome. Now, again, kind of reviewing this concept of genetics, some of the vocabulary we keep hearing right now, and I have mentioned it before, is the word genomics. So genomics is basically us comparing and assessing or analyzing the function of many genes, what those genes basically do in our body. Why do I have that gene in the first place? Why is it active in some cells and it's not active on the other cells? And again, because genetic is such a common thing that we use in a day-to-day life, we need to understand the potential dangers of us using genetics on our day-to-day basis, applying to medical technology, using it for modification of our food, modification of our offspring. So there is a lot of controversies based on the personal view, religious view, culture, etc. So these concerns, these controversies can be addressed when we think about it in terms of bioethics, having conversation about what is basically right and wrong in terms of the genetic science. I just wanted to show you this picture. Again, I will discuss this in much more detail as we go through our future lectures. But again, giving you this idea of what the genetic information looks like inside the cell. So I wanted to make sure that this is understood, that on the top, we have the most basic level of our genetic information, which is basically a DNA as a double helix. So, sorry. So it's this pattern that you see right here, that again, forming this double helix coming down. And if I condense this, and again, this is different level of condensation. If I really condense this DNA, I create what I identified as my chromosomes, which are these X's inside the cell at very special occasions, right? They're not always super condensed. We usually have the DNA slightly condensed, but not as super condensed as chromosomes. And this whole structure will be representative of our cell. As I will go through the future lectures again, we will expand on all of the structures and the condensation levels of DNA. Now, briefly, before we go into, again, going back to that genetics, I wanted to hint at what we're actually made up of, because it does help us understand why do we have the molecules, or why do we consume the food we consume. So what we have here is a word, macromolecules of life. So basically put is they are these gigantic molecules that are inside your cell. So if this is my cell, okay, within my cell, you have thousands upon thousands of what we call again macromolecules. And if you're not familiar with the word, macro basically means large. So basically, I'm saying that within your cell, you have these really large molecules that are responsible for specific functions. So one thing that I haven't listed here, and I think I will expand on it in the future lectures, all of these macromolecules are identified as organic. And this word has to be very particular. It's not what we use in our day-to-day shopping or grocery shopping, more specifically, when they use the word organic very commonly. The word organic implies that all of these molecules, they will contain carbon atoms, which is depicted with the letter C. And then they always also has H, which stands for hydrogen. So minimum, all of them will have carbon and hydrogen. Hence, we identified as carbon-based organism, and they're always going to, again, have hydrogen. They will also have other atoms like nitrogen or sulfur, but the most important building blocks of these macromolecules would be carbon and hydrogen, therefore making them organic. Now, what are these molecules? What is these macromolecules? We have carbohydrates, which in many cases we simplify to the word carbs. We have our fats and lipids. We have our proteins. And of course, nucleic acid, which is what this class is all about. So again, briefly talking about these four major categories of food that we have, or sorry, macromolecules we have. We have carbohydrates. The best example of it would be glucose. Sources of carbs are things like starch, rice, veggies, your fruits, right? All of these, bread, all of these are identified as carbohydrates. And for us, when we consume any of these, our body is going to break it down to glucose. It can digest it and break it down to glucose. And that glucose is basically what flows inside your body. It goes into your cells and are used as immediate source of energy. So if your cell needs to basically do anything, it will use glucose and convert that to the energy. And I'm going to introduce these three letters, and we'll talk about it again later on. The energy, the way that our body uses glucose is to convert it to ATP. Again, just kind of starting introducing the vocabulary for you right now. Now, fats and lipids, that's another category. And again, commonly, a lot of people, again, if you're watching a lot of videos, they try to scare you off of fats and lipids. But I do want to make sure you understand that fats and lipids are a good part of our life. We do need them in our life. It's just nothing in excessive. So for fats and lipids, think about your typical oils, right? That comes from plants like the avocado oil, grapeseed oil, canola oil, etc. And then you can also think about the fats that are coming from animals like butter or lard. Now, what our body actually convert these fats to are two major molecules, which is triglyceride and cholesterol. And all of these fats are used as long-term energy storage. So there's still our energy, these are main sources of energy, but the key difference is immediate for carbs versus long-term. So when you say you're going into fat burning, think about it that you don't immediately tap into it, you wait a little bit longer. Now, proteins, I gave you some examples like lipase, keratin, lactase. Basically, they carry all the functions in your body. They include things like enzyme, they give you your physical traits like your hair color, your eye color. They make you susceptible to being able to eat certain food and not eat some food. And they allow you to basically produce your energy or your ATP. They break down molecules, they put molecules together. So they do everything in your cells, everything in your body to keep you alive. Now, how do we make these proteins? How do we know what proteins to make and how much of it to make? For that, we're going to rely on our fourth group, which is the nucleic acid. Nucleic acid are identified as things like DNA and RNA, again, common terms that you may have heard. And these are the ones that store our genetic information. And this is what this class is going to be about. How this DNA and RNA is stored, how those changes in that going to affect our proteins because they're directly related to each other. And again, how we can modify it to maybe benefit ourselves in terms of our food, in terms of our health, in terms of medication, etc. Now, since we are talking about genetics, we need to understand DNA. So DNA stands for deoxyribonucleic acid. So D, one part, oxy, ribonucleic acid. So the three words, the three letters come D, N, A. Now, what is DNA? So DNA structure, first of all, is a double helix. And I mentioned that earlier. Double helix, it means two strands come together to form this beautiful wrapped structure. Now, DNA, again, remember, it's a macromolecule. It's a really large molecule. So in reality, when we have a macromolecule, right, they get broken to a smaller molecules, or what we call our subunits. Basically, what is made up of, right? So the subunits of our DNA, our nucleic acid, are thing called nucleotides. So nucleotides are very important for us because they're the way they're set up together, the way that they're held together, basically form your DNA, which makes your genes, which makes the proteins, which makes you basically who you are. So what are my components of nucleotides? And again, I will expand it. We're just introducing. We have what we call a phosphate piece. We have a 5-carbon sugar. And then we have one of these bases that I have here. So we have a base thymine, which is represented with letter T, cytosine, they're represented with letter C, adenine with the letter A, and guanine that's represented with letter G. So 3 parts. So we have a phosphate, a sugar, and what we call a base. So this is the options that we have, which gives us our DNA. Now, I will break down the word DNA a little bit more in detail as we go through the PowerPoints. Now, as I mentioned earlier, the double helix structure of DNA is very interesting. It's very important and is extremely, I guess it's extremely stable in terms of having those things and very particular in terms of how they're matching together. So if you look at your double helix, so this is on the image, you can see your double helix over here. And on this page, I kind of unwind your double helix. One thing that you notice, if you look at the letters, your A is always base pairing with the letter T, and G is always base pairing with C. So this is always going to happen. It's very important for you guys to note that. One other thing that I want to kind of, again, hint at right now before we go into detail about them is that these dots that you see right here represent what we call hydrogen bond. They're the one that hold these double helix together. And again, one thing that I do want to note, and it will be hinted again later on, is that A and T are held together with 2 hydrogen bonds, and C and G are held together by 3 hydrogen bonds. So it's harder to separate C and G from each other versus A and T. Now, the structure of the DNA gives us the ability to basically create life, essential for our ability to continue creating the offspring and passing that genetic information forward. The essential abilities, again, one is that it can replicate itself, which means, again, you can pass that genetic information further on to the next generation, or you can replicate the cell by using that genetic information. And using that genetic information, you can synthesize your proteins, which are basically your workers, right? These are your workers. They are the one that carry functions inside your cell. So you have to remember that. I'm going to keep reminding you of that. Now, again, going from the gene to protein, looking at some of the vocabulary, not exactly the process, but rather than vocabulary. So when we're looking at the DNA replication, we're starting with our double helix. Again, that kind of a pattern of double helix that is important for the structure of the DNA. And when that the double helix is done, what I will be creating is two double helix, if I can actually draw a double helix. Sometimes it doesn't work as easily. And these are referred to as the DNA is referred to as the daughter DNA or the next generation DNA. And that DNA is basically used, duplicated when the cell is undergoing cell division, which basically means if you have this genetic information for one cell and you want to make it 2 cells, you have to make the same copy for each cells. Now, DNA replication happens when the cell is undergoing the cell division process. And again, we'll expand on this later on. So this part, the replication of the genetic information is associated with cell division, and it could be a process called mitosis or a process called meiosis. But what if I want to use my DNA to make my proteins? So how does that work? Now, here's the important thing. you remember that DNA is extremely important and also extremely fragile. So how do I make sure that I do not damage my DNA? Because if I damage this DNA during the process of protein synthesis, I basically lose the ability to synthesize new proteins because I only have one copy within that cell. So how do I protect that DNA and still utilize it to produce new proteins? And what I do is I create something called RNA. And DNA basically makes copies of itself and creates what we call ribonucleic acid or RNA. And again, see the similarities in terms of name. Nucleic acid, the word ribo is repeated. So it's basically very similar to structure of DNA. Now, this process of copying DNA to RNA is referred to as transcription. So transcription is basically, again, making DNA copies to form RNA. The process also has another name called gene expression. So if you see that, it's basically the same terminology. Now, this is RNA. Again, we're not at the protein level. So how do you take the RNA and use it to make your proteins? Now, that process is known as translation. So translation, again, utilizing the resources inside the cell, including what we call ribosome, an organelle called ribosome, to use the RNA and synthesize your protein. And proteins are basically, again, going to be your workers. They're going to give you your traits, the characteristics, conditions, susceptibility to medications, etc. So here is a depiction of what I was trying to say. So right here, you have the DNA. And notice that there is an arrow that's kind of reverted back to the DNA. And that implies that DNA is replicating or copying itself. If I'm going from DNA to RNA, as you can see it here, the process is known as transcription. And transcription, you make your RNA. Now, by just looking at this, one thing that you should notice about DNA and RNA is, again, DNA is your double helix and RNA is a single strand. So please do note that as we will follow up on this concept later on. And later on, again, the process is utilizing the RNA to make your protein through a process called translation. During this process, RNA is not used. So RNA is not used is basically a blueprint to produce the proteins, but itself, it doesn't get utilized throughout the process. So by the end, when you're done making your protein, the RNA is still there. So what happens if there is mutation or changes in your DNA? If that happens, or sorry, I should say, if there is changes in the sequencing of your DNA, in that case, we refer to the word as, or the process as mutation. So any changes in the sequence of your gene is referred to as mutation. Please do note that some of these mutations may be very dangerous and can cause significant harm to the person and even lead to death. And sometimes this mutation has zero effect on the person. And again, so those are basically, if it has no effect, it would be called neutral. If it causes damage or is detrimental to the health, it's called negative. And sometimes they actually make the gene even better than it was before, and we refer to those as positive mutations. Now, mutation in either sperm or egg can pass that problem or that mutation or the changes to the next generations. And again, those changes can be either have no effect, can be negatively impacting the offspring, or improve the function of the gene / protein. I do want to mention, maybe some of you guys have heard of this medical condition called cystic fibrosis. This is a very good example and a common example used to describe a negative mutation. So cystic fibrosis, shorthand to CF, is a medical condition that's created by a mutation in a gene called CFTR. And then CFTR is just basically a membrane protein. So transmembrane is a protein that's inside the membrane, and it basically regulates what certain thing gets into your and out of your cell. So as you can see, it's a selective doorway. This type of genes are very important in our airways. So they're affecting your respiratory system in another sense. So what happens when there is this mutation that leads to cystic fibrosis? You are replacing a single amino acid that is called glycine. And again, you don't need to know the name of the individual amino acids, but in this case, it's a very specific change in the amino acid from glycine to what we call aspartate or aspartic acid. If you don't know what amino acids are, again, if you think about your proteins, protein is a macromolecule. So it means it has to have individual subunits, and the subunits are called amino acids, or AA for my shorthand. So each of the circles I'm drawing is an individual amino acid, and the whole structure would be your protein. So basically think about, you take this amino acid, let's say this was originally glycine, and you change it to aspartic acid. And this change would literally cause the medical condition. So what is this? When this protein changes, it means that it doesn't allow the exchange of basically your door gets shut down. So the selective doorway we mentioned up there, it gets closed off. Now that leads to thickening of the secretions that are produced in the respiratory system. It's going to affect the breathing because it creates this very thick mucus that lines the respiratory path. It can affect the digestive system because the same type of cells are involved in the process of digestion, and you have other symptoms as well. This image depicts what that change looks like. So all you're doing is, in the DNA level, you're changing a C to a G. Sorry, no, you're not saying I apologize. You're changing a C to a T. I apologize for coughing. So that change leads to a change in the RNA. So the RNA goes from a G to an A. So that change happens, which means now the protein, this amino acid, this is your amino acid, goes from from glycine to aspartic acid. And instead of this channel being open, it's now closed. Here again is some of the main parts of the body that gets affected. So sinuses and airway are part of your respiratory system. You have effect on liver and pancreas, which is part of and intestines that are part of your digestive system. Reproductive tract is affected, and then the skin is also affected by producing what we call salty sweat. So here is what we already mentioned previously, but reviewing it in a little bit more detail. So we learned that the chromosome are these egg-shaped structures that are basically the super condensed version of our DNA. Now, one way for us to grab this DNA is around these proteins. And I won't mention the proteins, but again, I just want to kind of introduce the name of the protein. The protein is called a histone. It's a circular protein, and kind of like your DNA is wrapped around it. Sorry, it's not the best picture. Now, if we look at the cells that we have in our body, we actually have two types of cells. Most of our cells are considered somatic cells, which basically cells that form every part of our body with the exception of our sex cells, which again, if you're a female, it would be your oocyte or your egg, and in male would be the sperm. So somatic cells would be skin cells, again, examples, gastric cells, gastric and cardiac. Gastric basically means your digestive system, more specifically stomach. Cardiac means your heart. So these are cells that basically form your body. We mentioned that there are 23 pairs of chromosome. Again, 23 from your mom, 23 from your dad. And out of those 23, 22 of them are identified as autosomes, which basically means they are looking at characterizing anything that is shared between male and female. So there's no specific characteristics. They're all encoding for creating a human being, basically. And then the last of those 23 pairs, the last pair is what we call sex chromosomes. If each parent gets passed down one of those chromosomes to their next generation. So if you have a male, it would be XY, and the mom is always XX. And again, we're talking about under normal condition. So the offspring is going to get one copy from each side. So if dad contributes an X, mom is also going to contribute an X because there's no other option. So you would get a girl. And if the dad contributes a Y and mom contributes an X, then it would be a boy. So the Y is what makes somebody a boy. Again, there are mutations where you would get XXY or XYY. That again, this is the abnormal aspect of the genetics, which we'll discuss in more detail. But males will always have X and Y if you're a healthy male, and female will have an XX, one from each parent. Here's a depiction of what those chromosomes look like. Notice as we go through the numbers, the size of these chromosomes do decrease. And in this case, they're depicting a female because you're depicting an X and Y. FYI, there are a lot of what we call sex-linked genetic disease, and they're all associated with the X chromosome. Now, the word traits is something else that you may hear. A trait caused by a single gene in terms of Mendelian genetics. And in that case, we use what we call a dominant versus a recessive gene. So dominant gene versus recessive gene means if you have the dominant gene present, it's going to take over and you will see the traits or the physical characteristic associated with the dominant gene. And if you get two copies of the recessive gene, that's when you will see the recessive characteristic being visualized. And again, I'll give you an example in just a minute. So here is Mendelian genetics. If you have the dominant traits, you will have freckles. So if you have one copy, regardless of from your mom or dad, or two copies, you will get freckles. So you can have two copies or one copy of the gene, and you still would have the characteristic. If you have 0 copies, then you will have freckles. But only, again, one copy is enough for you to develop that trait. Widow's peak is what I mentioned before, presence of it versus absence. Again, you need to have one gene or two, and you will develop it, versus if you don't have it, so neither of the parents pass it on to you, then you will not have it. And then the earlobe, free earlobe versus attached earlobe. This is also, again, single copy will be sufficient to pass this trait forward. Now, what I mentioned earlier, we talked about Mendelian, which is, again, kind of think about it as black and white, if you want to say. versus complex trait. Complex trait, as the name implies, you have multiple genes are involved and also environmental factors. So both of these are going to contribute to how those traits will look like in an individual. So for instance, if you are looking at our examples here, skin tone is a really good example. Both the environment, if you're looking in kind of in a sunny environment versus a really kind of, I guess, less sunny environment. Eye color is a big contributor. The height and weight. So height and weight, eye color, and skin tone are all come from not only one gene, you have multiple genes involved, but also, again, environmental factor play a big deal. This makes it very difficult to figure out how those traits will be inherited and how they will represent themselves in your offspring, because there is multiple factors that come into effect. One of the examples I was tell my students is breast cancer. So having a gene does not necessarily mean you will develop it, because there are things like environmental factors that could definitely impact if you will develop that cancer or not. Now, since we are talking about genetics, it's also good to understand how the levels of complexity works in our body. Even though I use the word genetics here, in truth, it's level of organization for us as humans or for any complex organism. So we start at the most basic level, which is referred to as atoms. If you have ever seen a periodic table, you probably have heard of atoms. So some of the examples are things like oxygen, carbon, hydrogen, nitrogen, sulfur, etc. They can put multiple atoms together to get your molecule. So in this case, they put 2 oxygen atoms together to get your oxygen molecule. Those molecules form your macromolecules, which we already talked about in detail. Things like your carbohydrates, proteins, nucleic acid, fats, or lipids. All of these macromolecules are inside your cell. I mentioned this before, but now we have a visual representation of it. Cell forms your tissue. And I'll talk about that in just either in this lecture or future lectures. There are 4 major types of tissues we have in our body. Multiple tissues give rise to our organs, like your lung, heart, kidney, liver, intestines, which gives rise to your organ systems, like respiratory system, digestive system, cardiovascular system, nervous system. That gives rise to the organism, which in this case, they show a lion, but we're talking about humans. And then if you look at this, human gives rise to families, and families give rise to populations. And when we're talking about the evolution and the changing, we want to think about evolution of a species. So humans are a species called Homo sapiens. So we will look at the species as they evolve. And remember, again, the evolution always starts at the most basic level and then work its way throughout all the way to the population. Now, again, when we talk about the cell, so I draw this kind of like a circle before and then nucleus inside of it, and then you had the X's that represents the chromosome. But in reality, if you're looking at the cells inside the human body, each of these cells have their own shape based on where they're located, what's their major function inside the body, to kind of give you a clue what these cells would look like or visualization of it. We have things like muscle cells. So this is a smooth muscle. We have depiction of fat cells, which is also known as adipocytes. We have a bone cell, which basically keeps your bone alive and healthy. blood cells. In this case, they're depicting red blood cells, a nerve cell, basically what your brain, spinal cord's made up of. Epithelial cells, in this example, they give you things like your skin, immune cells like white blood cells, like neutrophils, eosinophils, and then sex cells. So you have your egg and you have your sperm. So again, variation on the cell is very obvious, but it's very interesting to know that even though these cells are very different, they do share, and again, there's exceptions, they do share all of the internal structures. And these internal structures are basically what keep the cell alive. Now, what I want you guys to understand is at the center of this, we have what we call stem cells. And I think, again, if you listen to news, if you follow any particular news channels that are associated or talk about genetics, you have heard of stem cells and the controversies behind the stem cells. So we will discuss that again in more detail. Now, we are going to use stem cells, and we're going to undergo a process called differentiation. So differentiation is basically changes that happens inside the cell that makes them specialized in a very specific characteristics. So in this case, we start again with the stem cell, which is the main cell, and each of them differentiated to the cells that you see. So they become specialized, they get trained and transformed to carry a very specific function within your body. So again, when you look at the stem cells, it's important to understand, and I want to kind of make sure that this is understood. So if I have a stem cell, you can replicate that stem cell. So kind of go through the cell division and create new stem cells, or you can take this path and undergo differentiation to make variations of the cells that are needed by your body. So you either, again, replicate yourself or you differentiate the stem cell to create various type of cells with a specific function within the body. This picture is depicting the type of tissues we have in our body. Again, it's important to understand that tissues are basically made up of cells, hence we're talking about them. We have 4 categories of tissue. So I want to make sure that, again, this is highlighted for you here. We have muscle cells with 3 subcategories: cardiac, skeletal, smooth. We have epithelial, which is a total of 7 different types. There's only 4 of them listed here. Nervous tissue, which is the easiest one because you think about basically anything that has to do with your nervous system. And connective tissue, that gets really complicated, so I'm not really going to go into details. But what falls under connective tissue are things like tendons, ligaments, bones, fat cells, blood. All of these fall underneath your connective tissue because it holds your body together and basically keep everything working. This picture is depicting the common organs you have in your body, again, kind of working through those organizational levels. I like this picture because it also works as a way for you to understand the anatomy of the body a little bit better. So the location of things like your liver, your stomach, your pancreas, gallbladder, a small intestine, large intestine, the reproductive organs, kidneys, etc. And again, organs give rise to what they call organ systems. Things like cardiovascular system, so cardiovascular system, respiratory system, digestive system, nervous system, what we call lymphatic system, which is your immune system, and things like endocrine, etc. So again, the goal is for you guys to understand the complexity of the levels of organization. So now that we have a basic understanding of some of the terminology, let's look at some of the applications of the science of genetics and genomics. So as I mentioned multiple times already, genetics has become part of who we are. It's literally have created its own space inside our life. It affects our health. It affects our what we eat. It gives us information about our past and guidelines to our future, creating our offspring, make sure they are good, make sure they're healthy, certain characteristics to be developed, etc. So citizen scientists are basically identifying, discovering this huge amount of genetic information that's basically helping researchers compile what we call genomic databases. So we literally have people coming and sharing their genetic information, having that, allowing that to be sequenced and figure out what is the genome of each of these individuals are. And understand, guys, the more we know about our genome, the more we will be able to understand who we are and again, help resolve possible medical condition, find cures for disease, and make sure that the next generation is protected and have the best chance of basically surviving. So as I mentioned again, think about bioethics. If you think about genetics and what power lies within genetics, many people fear it, especially if they don't understand it. For many people, it provides hope because it helps them find cure to their condition. Sometimes people get angry because it goes against what their personal belief or religious belief are. Wonder, the magic, the power behind it, what we can accomplish, despair, and depending on the basically what you do, right? Sometimes we just get sad as why you can't do more, right? And it is very important to understand that you need to learn and comprehend the science before you make decisions, before you fear something, before you get angry with something, right? So the goal of this class is for you guys to understand what genetic is so you can make informed decisions, so you can inform your parents, you can inform your kids and your partner about what genetic is and use that knowledge to basically make a better community. Something else that I want to kind of finish up with, I guess we're getting toward the end of this PowerPoint, is establishing the identity. So allowing to compare our genetic sequences, our DNA, to one person to another, help us basically identify who we are. This is, again, really good, important in terms of CSI and forensics. It can help us figure out our relationships, who we are related. Like if you have half brothers or half sisters, who's your mom or who's your dad, and then ancestry, where you come from, right? Kind of think about again that genealogy concept. The DNA profiling is another thing that is again has become such a common thing in terms of forensic science, right? If somebody gets killed, if somebody gets sexual assault, right? All of these can be basically helped. Those cases, those forensic cases can be helped by using genetic information. Identification of victims, if they are victims of natural disasters, finding better food, right? Creating GMO food or genetically modified food, or even identifying if the food you're eating is authentic. So things like caviar, are they actually coming from fishes that have that lineage that give you that good high quality food? And then again, it's important to understand that when we looking at that genetic information, when we are tracking it, when we kind of go back, we can figure out where we came from, right? We can figure out the what we already found in other sciences and provide additional support to that. So it's very important for sciences like anthropology, right? Finding things that may contradict each other, but that DNA can help us figure out what is the correct answer. DNA testing can also help us, which is I think really fine, help us basically analyze the mummies in the Egypt kingdom and figure out what was the reason that they died. So like this is really fun. They found out that one of the kings actually died from complication from malaria, and that was pretty interesting. And this is just basically studying DNA, right? Having that tool at the extra tool. Or another cool thing about, again, history, thinking Thomas Jefferson, our president, our former president, or many years ago, had children with one of his slaves, and now they have this really extended family that actually form and uh, uh, they actually meet every year, and they take pictures and they are included, and it's nice to see that diversity. And the only way it was possible for these people to figure out that they are related is by looking at the genetic sequencing. Another aspect of genetics is conservation genetics. So again, the word conservation has been thrown out a lot in the recent years, but it's important to see and assess it in terms of genetics. So what you see in this picture is what we call a white rhino of Africa. They are they were heading for extinctions. There were very few of them left, and they were able to combine the genetic and reproductive technology to create and rebuild this population, which is, I think, is absolutely amazing. So you're bringing a species that were very close to extinction, you're bringing them back, and you're helping develop them and basically grow that population. Researchers are also working on bringing back and the species by borrowing the genetic information, the genome of other species that are related, what we call subspecies, basically. Precision medicine. This is one of my favorite topics. So when people are volunteered to their genomic sequences, which is what I mentioned before, the more data we collect, the better we can understand how diseases are developed. We can better understand the history of the changes that happens in a genome that leads to those medical conditions. So these databases, these gigantic databases, allow us to create medication that are very precise, that are tailored to every single individual based on their genetic information, rather than kind of hoping that this generic medication we give are going to work for everybody. So we actually have a branch of science called pharmacology genetics, which is pharmacogenetics, which is basically using a specific drug that's going to have a kind of personalized to that individual and limit the side effects. So you're gonna basically have a medication that is very targeted with minimum side effects. This is something, again, I mentioned a few times, gene modification in terms of the food that we eat. I like this chart. You guys are welcome to read over this in a little bit more detail. But the idea is, again, we are genetically modifying our food, get it be vegetable or animals, to either help them deal with pests, make them larger to make sure that they provide for larger population, make sure that they don't go rotten really fast or spoil, adding vitamins and minerals that may not be naturally found in them for third world countries that use that as a food resource. So you may hear the GMO as a very negative concept, depending on who you listen to, but there are a lot of benefits also to anything that's genetically modified to help us basically improve our life. So going back to that whole idea of the genetic modification, I want to point out that uh, we use a technology called recombinant DNA technology. And again, I will expand on this later on. We're just introducing the word right now. So in this case, what you see is they take the gene for human insulin um, out of the regular human genome. They put it inside the E. coli. So this is the genetic information of an E. coli. They literally put the human insulin gene inside the E. coli genome. And E. coli, if you don't know, it's a bacteria. And then they grow and grow and then grow on E. coli. Lots of E. coli. And remember, because bacteria are very easy to grow, not like humans, right? Uh, they basically grow and double in numbers in every 20 minutes. So you can grow this really, really easy by doing very large scale process called fermentation. So now these E. coli have produced the insulin as one of their proteins. You can isolate and purify that insulin and then basically put it in the pens for people who are diabetic to inject themselves and make sure that they are maintaining their glucose. Genome editing is another thing. It can either replace, remove, or add a specific gene to any organism. So again, what we did here, that genome editing in this case was addition of the insulin gene of a human to an E. coli bacteria. Now, if you guys recall the word exome, we mentioned that before, is basically all of the exons in a genome, right? That's called the exome. So exons are important because it encodes for proteins, right? So those are the parts of the gene that actually encodes for proteins. So if you are sequencing these giant exomes that basically, again, give us our protein, we have this really large database of information, right? And we can look at the variation in the genes. We can look at how those variation lead to physical differences from one person to another, how those variation may lead to disease, right? So finding this information basically help you advance and form connections to how a gene change or modification can lead to a condition or a specific characteristics. So if we later want to develop that characteristic, what type of exome should we look at? It's also very, very important for us to use that exome genetic to identify very rare medical condition in a very rapid process by just doing a sequencing. So is it important for us to do these processes? Yes. Before, and again, because the technology is advancing so rapidly, processes that used to take years and years to complete now takes matter of days and or sometimes hours. So which is really, again, cool for us. Now, because we're sequencing genomes and this is basically affecting every person in every part of the world, and also a lot of the species and animals. Think about, again, those kind of extinct animals that may come back using this technology. We do need to understand that it's not only impacting us humans, it's impacting the whole Earth, right? Because we share our planet with all of those species. So we have this science, which I think is also very, very cool, called metagenomics. Metagenomics, again, think about meta as this really, really big, large field. This is involves sequencing of DNAs in a habitat. So think about an environment. You have maybe hundreds, maybe thousands of species in that. So megagenomics literally goes into that habitat, goes into that environment, and takes the genomic sequence of every animal, from bacteria to fishes to birds to larger animal, larger vertebrates, snails, etc. the first metagenomics project was done on Sargasso Sea, which was really interesting because they literally sequence every genome with a genome of every species within that environment. Here is what it looks like. You don't need to understand what it is, but I think it's really cool for us to understand the number of the species, and each of those little lines or little red lines or the green lines or the blue lines are identifying a different species or subspecies that are found in that environment, the Sargasso Sea. But why do we care this? One of the things that we can learn is how these species actually interact with each other. How are they related to each other? And in many cases, we can actually use that information to figure out new drugs, new medication, new energy sources, things that we may not have been aware before that lives in an environment may help us develop new ways of basically extracting information, getting new drugs, and basically maybe even help a species grow and develop that might have been possibly close to extinction. Now, we do need to also understand that we are trying to look at this global aspect of it because it affects a lot of our social issues that will go parallel to our scientific progress. So does everybody will have, as time passes by, does every human have the same access to this genetic information? Does everybody have access to the same treatments that are genetically based by doing modification in the gene sequencing and forming those individualized drugs and medication? How can we possibly misuse that information? Again, kind of think about culture or think about mad scientists, right? Things that people that want to do that not ethically sound correct to many, many people. Can you abuse that information and actually cause harm? For instance, can you make drugs or poisons that are specific to individuals and doesn't affect the rest of the people? And could you possibly use that as a tool so that something may harm an individual or a specific population? Again, we'll expand on that later. It is a very fast moving science. And again, there is a lot of ethical issues that are associated with this, but it also has a significant amount of potential. It can become something that can help many lives and many species. As time passes by and as we get better familiarizing ourselves with the genetic and the science behind it, every nation develop their own guidelines of how that genetic information is being used, spread, and what they can actually utilize by keeping in mind the bioethic components. So bioethics, again, is going to be a really, really big discussion and may vary from country to country based on their culture, belief, religions, etc. So that's the end of our first presentation. I just wanted to make sure that you guys, if you have any questions, please contact me. You're more than welcome to email me and ask your questions. Make sure you guys complete your Canvas posts that are associated with this lecture. And then we'll go into our next lecture that is introducing the concept of scientific method. And thank you so much, everyone.